Good morning, dear students. This is your biology teacher, Miss Nidal. Today, Wednesday, March 11, we're going to start with a new part, immunology. It consists of 3 chapters, 6, 7, and 8. Today, we're going to start with Chapter 6, Role and Components of the Immune System. Document 1, Hla and Major Self Markers. So please open your book on page 114. What is immunology? It's the science that studies the immunity. Immunity, it's the defense of the body against invaders like viruses, bacteria. As you know, each function in your body is ensured by a system. Thus, immunity is ensured by an immune system. And like any system, it's made up of organs, cells, and molecules that are responsible for this immunity. What are the objectives of our document? The first one is to define HLA or MHC, and the second one is to explain how the immune system distinguishes between the self and the non-self. Document 1, HLA and major self markers. What is the self? The self is the set of molecules expressed on the membrane of cells and specific to each individual. So, it is what belongs to the body and tolerated or accepted by the immune system. However, the non-self is what doesn't belong to the body, and thus it should be rejected by the immune system like virus and bacteria, etc. We're going to talk more about the non-self in document 3. So, in this document, we're going to answer the following question. How is our organism able to distinguish the cells of the organism self from the cells that don't belong to the body and that represent the non-self? The immune system distinguishes self from non-self through the self marker that is called MHC or HLA. HLA, also called human leukocyte antigen, they are proteins located on all nucleated cells of the body and that characterize the genetic identity of each individual. They are also called self markers. They are coded by genes located on chromosome number 6. They are also known as MHC, major histocompatibility complex. Why? Since they help to study the compatibility between the tissues of the donor and those of the recipient in transplantations or grafts. Look please at document C on page 115. This is chromosome 6, where are represented the 6 HLA genes ABC, GP, DQ, DR. These genes are divided into 2 groups, HLA class 1, HLA-A, HLA-B, HLA-C, and HLA class 2, GP, DQ, DR. HLA class 1, they code for HLA antigens or proteins located on all nucleated cells, while HLA class 2, they code for antigens or molecules that are located only on some immune cells like the macrophage. Here is an example of a skin cell and an immune cell like macrophage. The skin cell represents on its surface only the HLA class 1, while the immune cell represents on its cell membrane both types, HLA class 1 and HLA class 2. The uniqueness of the biological identity of each individual is so related to the polymorphism in the HLA molecules. Why? Since there are 6 HLA genes, A, B, C, GP, DQ, DR, and every gene has several alleles, moreover, these alleles are codominant, so these make us unique. Of course, in some cases, like in the case of identical twins, they present the same HLA molecules. Also, there is 25% probability for sisters and brothers to have the same HLA. So, the immune system recognizes and tolerates the cells carrying the self HLA, but it rejects the cells carrying the non-self HLA. To better understand how the immune system differentiates between self and non-self, these experiments are performed. Please look at document A, page 114. 3 types of grafts are considered here. Autograft, which is the transfer of organs from a part to another in the same body. Isograft, the transfer occurs between two genetically identical individuals, and allograft, where the transfer occurs between two genetically different individuals. The aspects of the graft are observed 2 days later, 1 week, and 15 days later. Dear students, it's better if you stop watching the video for a while and try to answer my question. Interpret these experiments. So, how to do an interpretation of these experiments? The aspects of the graft are the same 2 days later. Vascularization appearing around the graft with a pink color in the 3 cases of grafts. However, 1 week later, the graft is integrated into neighboring cells only in the case of autograft and isograft. However, in the case of allograft, where the individuals are genetically different, there is redness and edema around the graft. This shows that the reaction of the organism appears starting the 7th day. Moreover, 15 days later, the graft is accepted only in the cases of autograft and isograft, while it's rejected in the case of allograft where the two individuals are genetically different. This shows that the rejection occurs within 15 days and the success of the graft happens only when the two individuals are identical or when it's done in the same body. Please look at document B, page 114. This document represents the results of 2 studies concerning the compatibility of kidney transplant between donor and recipient. What is incompatibility? It means no similarity. What means the incompatibility number is 0 between HLA of the donor and HLA of the recipient? It means that the compatibility between HLA of the donor and HLA of the recipient is 100%, or that the HLA of the donor and the HLA of the recipient are 100% similar. I also prefer here you stop watching the video for a while, for a few minutes, and then try to answer this question. Interpret the results shown in this graph. How to do the interpretation? The study performed by Eurotransplant shows that when the incompatibility number is 0, the survival of the transplant is 72%. However, it decreases to 60% when the incompatibility number increases to 4. This is for the Eurotransplant. Similarly, the study performed by Oxford shows that the percentage of graft survival is 85% when the incompatibility number is 0 concerning HLA-DR. However, when this number increases to 2, the percentage of graft survival decreases considerably and reaches 55%. Then, what does this mean? This means that the increase in the incompatibility of HLA between the donor and the recipient, especially regarding HLA-DR, it reduces the percentage of success of the graft survival. As a conclusion from documents A and B, the graft succeeds 100% when it occurs between individuals having similar or compatible HLA, and the graft is more likely to be accepted if both individuals, the donor and the recipient, have a higher number of compatibility at the level of HLA molecules as in the autograft and isograft. To finish this document, let's see what is the immunological self. The immunological self is the complex resulting from the association of the peptide molecules that result from the hydrolysis of protein molecules to HLA molecules on the cell surface. All normal body cells synthesize self-peptides that are displayed on the cell membrane bound to HLA. The complex HLA self-peptide, it represents the immunological self that is tolerated by the immune system, and it helps it to differentiate between the self and the intruders. This figure will help you to better understand what is the immunological self. Normally, in every cell, samples of self-proteins are produced by the cell, like by transcription and translation. These self-proteins are fragmented into self-peptides. And as you see here, these self-peptides, they bind to the HLA class 1, and together they form the HLA class 1 peptide complex. This complex will be displayed on the surface of the cell and thus form the immunological self. So this immunological self will be well tolerated by the immune system. Thank you for your attention.